Hello, everyone. I am Shaojun Pan from Fudan University, China. Today I want to talk about our recently proposed tool, SemiBin, a Metagenomics binding tool that incorporates information from reference genomes with semi-supervised deep sequence neural network. Metagenomic sequencing is commonly used to study microorganisms in the environment without a need for culturing. The standard approach of analyzing metagenomic state is to first assemble short reads into longer contigs and then use we call binning and grouping the contigs into bins. And each bin is predicted to contain contigs from the same genome. For now, binning methods can be divided into two categories, depending on whether they rely on pre-existing genomes called reference-dependent or operate novo called reference-independent. Reference-independent methods can recover completely novel species, so it is most useful in the recently metagenomic research. Most reference-independent methods rely on sequence features like chimeras and the abundance features. One exception for now is SemiBin, that the only existing semi-supervised binning methods taking advantage of mass-link and canonical constraints from additional information, such as taxonomic assignments of contigs. So here we introduce SemiBin, a novel semi-supervised metagenomic binding tool. So here we proposed a better way to generate reliable mass-link and canonical constraints from reference genomes, and we used a semi-supervised neural network to incorporate this information. And also we have evaluated that SemiBin can outperform existing state-of-the-art methods in simulated and real datasets. And we will talk about these details later. The first thing, the mass-link and the canonical constraints is an idea used in we call semi-supervised clustering. In our binning question, a mass-link constraint between two contigs means that two contigs will be in the same genome, and the canonical constraint means that two contigs will be in the different genomes. When generating the constraints from the reference genomes, here we use the way that generate a canonical link from reference genomes if they have different labels and for mass-link constraints, considering the noise of the annotation. So here we break up long contigs to get a mass-link pair. So then the mass-link and the canonical link constraints are as inputs to the deep learning model later. So after generating the constraints, we used a semi-supervised neural network to learn information from them. So as shown here, the semi-supervised neural network is two neural networks with shared weights. The inputs to the model is the mass-link and the canonical link constraints generated before. And the purpose of the model is to use neural network to learn a better embedding that makes the distance between mass-link pairs small and the canonical link pairs large. So after the training, we will have a better embedding of the underlying structure of the environment, and these embeddings will be used in the clustering later. So this is a whole pipeline of SemiBin. The inputs to the SemiBin are the contigs and the BAM files from the mapping. So first, SemiBin generates mass-link constraints and the canonical link constraints as discussed before. And the second, SemiBin calculating the camera frequencies and the abundance features that are also used in other tools. So then SemiBin trained the semi-supervised neural network with the mass-link constraints and the canonical link constraints as inputs. In the next part, if the number of samples used in here smaller than 5, SemiBin assumed the number of reads per base, a base normal distribution, and calculates the Gaussian level of divergency as abundance similarity between contigs. Then SemiBin generates a graph with the embedding. Later, a community detection algorithm is used to get the binning results. In the binning project, in the binning methods now, so there are different binning modes used in the metagenomic binning. So for example, like single sample binning, co-assembly binning, and multi-sample binning. So single sample binning means that binning each sample into genomes after independent assembly, and it can be run parallel and very fast. Co-assembly binning means that samples are co-assembly co-assembled first and then binding contigs with abundant information across samples. And multi-sample binding means that beans are sample-specific and abundant information and is across samples. Here, SemiBin supports all these binding modes, and we benchmarked the SemiBin in all these binding modes. So here are the results. So first, we benchmark SemiBin in the simulated datasets from Karmi challenge. So in the Karmi datasets, SemiBin outperformed existing binders. In Karmi 1 simulated datasets, SemiBin returned more high-quality bins, and in the Karmi 2 datasets, SemiBin reconstructed a larger number of distinct high-quality genera, species, and strains. Also, in the Karmi 2 datasets, SemiBin can reconstruct more strains for the same species. Then we applied SemiBin to the real datasets. The default pipeline of SemiBin is to train a model and then bring the contigs for every sample. So considering the computational time usage when annotating contigs and training the model, we tried an idea that trained a model from several samples and applied it to other samples. So here is the result. We have found that model trained from more samples performed better, and when enough samples are used, it outperforms the default SemiBin and also the MATBAT2. I think the best tool that used before that used in the existing metagenomic research. So here we termed these versions as SemiBin pre-train. So train a model from one sample or several samples set, and then directly use this model to other samples. With SemiBin pre-train, so we do not need annotate and train model for every sample. It means that we can use SemiBin in very large-scale metagenomic analysis. Then we benchmark the SemiBin pre-train from 20 samples in the benchmarking of real datasets with single sample binding from human gut with 82 samples, dog gut with 129 samples, and ocean with 109 samples. In the left part of the picture, we have shown the number of high-quality bins, and in the right part of the picture, we have shown the difference between our other binaries compared to the SemiBin pre-train for every sample. So here, a negative point means that SemiBin pre-train performs better in this sample, and otherwise, a positive point means that SemiBin pre-train performs worse in this sample. So in single sample binding, SemiBin pre-train significantly reconstructed more high-quality bins compared to MaxBin2, VAM, and the MATBAT2 in the human gut, dog gut, and ocean datasets. Compared to MATBAT2, SemiBin pre-train has a 60.4%, 99.2%, and 48% improvement in the three datasets. When considering the results in each sample, SemiBin pre-train outperformed other single sample binders at most every sample. And also the p-value showed the statistically significant improvement. And we have also benchmarked the multi-sample binding, and you can find more results about that in our manuscript. When consider the overlap between the bins in the human gut for MATBAT2 and the SemiBin pre-train, most MATBAT2 generated high-quality bins corresponded to high-quality bins generated by SemiBin pre-train, and the SemiBin pre-train results further contained many high-quality bins which corresponded to lower quality or absent in the MATBAT2. Within bins that are present at high quality in the output of both binders, SemiBin pre-train achieved higher completeness and the F1 score, without a statistically significant increase in contamination. We annotated the high-quality bins from SemiBin pre-train and MATBAT2. We can see that SemiBin pre-train represents a larger taxonomic diversity at all levels, and the SemiBin pre-train was able to recover more genomes from both known and unknown species. So here, these results validate the ability of the semi-supervised Siemens neural network used in SemiBin to extract information from the background data, such as reference genomes, while being capable of going beyond it. So here in the summary, we proposed the SemiBin, a novel semi-supervised metagenomic binding tool. So in our experiments, we have evaluated that SemiBin outperformed existing binders in simulated and real datasets across 3 different environments. Also, SemiBin recovers a larger taxonomic diversity is a MVIF2, we think that it is very meaningful in the metagenomic research. And for now, SemiBin is available as an easy to install background tool. And here are some other links about SemiBin, so the GitHub and the BioArchive and the Anaconda. So another thing is that we will have an online SemiBin tutorial. So in the next month, so very welcome for your attention, and thanks for this paper's help about the tool and the manuscript, and thanks for listening.